Good afternoon. Thank you. I'd like to thank the organizing committee, Dr. Grimm, for inviting me to speak together, and I'd like to thank you all for showing up. I'd like to talk today about the concept of Cherenkov-specific contrast agents. I'd like to start by acknowledging the unique team of people that we had to put this together. It was basically a collaboration between two of our imaging cores, the optical core that I direct, and Julie Ciaprina is the technical director, and our nuclear subcore headed by Joel Karp. We had excellent chemistry support, both cold chemistry and hot chemistry, and as well as diffuse optical tomography support from Arjun Yode and radiology, Dan Prima. So as we've heard already this afternoon, Cherenkov radiation is the blue glow that you can see from a nuclear reactor, and it's the light that you observe when particles exceed the speed of light when traveling, charged particles exceed the speed of light traveling through the medium. From my perspective, I think there are three distinct advantages to why that I would want to use Cherenkov imaging. The first is that it gives you the ability to image a wider range of nuclei. The second is that you can use it in a high throughput fashion to screen various radioisotopes. And the third one is the concept that I'll talk about mostly today, that you can use it for functional imaging. Because it's an optical imaging technique, you can play this sort of standard tricks that people do in optical imaging. Photons can be quenched, they can be transferred to a longer wavelength, and the term has already been coined, Cherenkov resonance energy transfer, by Pivnica-Worms group at WashU. So there are, of course, a number of distinct disadvantages of Cherenkov imaging. We've heard about this in the first two talks. And the first one is the low intensity. The real big challenge is the low intensity and the low signal to noise, and that the wavelengths are really not suitable for optical imaging. We also get poor resolution for photon scattering, but I'm happy to say I learned something this afternoon. This is not as big a problem as I thought it was, thanks to the previous two talks. So some of the properties of Cherenkov imaging that are radiation that are particularly pertinent, again, they're emitted by all radioactive charged particles, whether they be positrons or electrons. The radiation is hyperspectral. It's a continuous emission spectrum, regardless of nuclide, and the signal intensity as a function is proportional to 1 over the wavelength squared. And the intensity of the light is given off as proportional to the beta particle endpoint energy. So I'll show a few examples of this. In the first one, we look at imaging electron emitters, and I've selected two examples from the literature here. And the first is RGD coupled to 90 yttrium and put into a mouse. And so this is actually the highest intensity Cherenkov emitter. And so you can image this. And this one is addition of 32 ATP into a mouse, showing that it goes to the bone, and you can measure uptake of ATP in the rib cage, and you can also see it in the brain. As I said, it's a hyperspectral emission, and it's continuous regardless of the nuclide. We've already seen this slide this afternoon, showing for a range of nuclei the emission spectrum. This over here shows work from our lab, just taking a phantom of F18, spherical phantom, and measuring it at a bunch of different wavelengths. And the line plotted there is 1 over lambda squared, showing that the intensity of the wavelength given off is approximately proportional to 1 over lambda squared. The intensity is proportional to the beta particle endpoint energy, and we've seen this slide in Dr. Cheng's talk. And again, F18 is a very good, one of the better nuclei for Cherenkov imaging, but by far, yttrium-90 has the highest endpoint energy and gives the highest signal to noise. And again, we can use it for high throughput imaging, and this is a recent example from the literature showing a yttrium-labeled probe linked to a molecule that forms disulfide bonds upon receptor binding. And so this is just an example showing that you can do 3 mice at a time on an IVA system. But for the main part of the talk, I want to talk about developing Cherenkov-specific contrast agents. And the main focus, the main point that I want you to take home today is that we believe that the true power of Cherenkov imaging will only be realized through the creation of specific contrast agents. Now, there's a number of ways you can do this, and in the previous slide, I showed you an example of conjugating a radioisotope to a targeted moiety, and Dr. Chen showed some examples of that as well. But the probes I want to talk about exploit the optical properties of Cherenkov emission, again, the transfer of photons or the quenching of photons through energy transfer. So I just wanted to point this out. This is not in any way saying that this is not a commercial for caliper in any way, but this is the system that we used. And I wanted to point out that on the caliper Lumina 2, they have 4 emission filters, and these are at those wavelengths, and I will talk about those wavelength bandwidths in more detail as we go. With a nice quote from Cherenkov at the bottom from his Nobel lecture, nowadays when researchers have at their disposal powerful sources of electrically charged particles and very specific measuring devices, measurements of this kind do not involve any special difficulties. So again, this is just an example of what the Cherenkov emission spectrum looks like when you look at those 4 spectral bandwidths, and this is just FDG diluted in a 96 well plate with the filters open, and then with the 4 emission filters that I showed in the previous slide. And as you would expect, you get the highest photon emission at the lower wavelengths because the emission is proportional to 1 over lambda squared, and as the wavelengths go up, the emission drops off. So we want to exploit this, and so we come up with this concept that we call selective bandwidth quenching. This is the theoretical 1 over lambda squared plot of relative intensity as a function of wavelength, and we want to design a contrast agent that will selectively remove an emission band from that continuous spectrum, and then we can do imaging at this wavelength or at more than one wavelength and add some kind of additional functionality to our imaging. This is not a new idea. Cherenkov himself recognized that quenching was possible. We can, for example, reduce brightness considerably, quench luminescence by heating it or adding materials which are able to quench the luminescence. So it's an old concept come new again. In the particular case of what we want to do, the requirements for selective bandwidth quenching are as follows. We need a chromophore that absorbs over a defined wavelength. We want that wavelength to be around 600 nanometers because we want it to be as close to the near infrared as possible, yet still have enough photon emission in order that we can see the photons being emitted. And then that chromophore also has to have the ability to report on a specific biological property, and that probe should be either activatable or it should be switchable. So to start this project, we started with the concept of working with pH selective agents. Many pH indicators absorb in the 500 to 600 nanometer bandwidth. The color change is reversible, indicating that a switchable agent might be possible. And of course, pH is very important in many aspects of biology. The pH of lysosomes sits around 5.5. External pH of tumors, extracellular pH in tumors is reduced, so it might have some kind of function in biology. And we're going to look at trying to make intermolecular and intramolecular pH sensors. So we started off with, we went back to high school chemistry, and we started with the pH indicator that everyone is most familiar with, which is phenolphthalein. It's shown in the acidic and basic forms here. It's a very commonly used pH indicator. It is well tolerated. It is the former active ingredient in the laxative X-Lax. It's not used anymore because at high doses it's suspected to cause cancer, but it is, we know that it's tolerable. It has a pKa of 9.2 and an absorption maximum of 564 nanometers. So we took sodium 18F labeled sodium fluoride, and we mixed it at varying concentrations with phenolphthalein and measured the emission at various bandwidths just using the IVIS scanner. And what you can see is that as a function of concentration, as a function of increasing concentrations of basic phenolphthalein, you see a quenching of the Cherenkov radiation when you leave the filters open, and you basically see that that quenching is bandwidth dependent. The lambda max is at 564, so you would expect the greatest bandwidth quenching to be in this range, and certainly that's what you see. This is a graphical representation of the integrated photons from this open one, and then at higher wavelengths where basic phenolphthalein doesn't absorb, you see less and less change. And so you can see that if you plot at each of the different wavelengths, you see the greatest change at 515 to 575, and when you get out to the longer wavelengths, you see absolutely no quenching of the Cherenkov radiation, but the initial emission is at a lower intensity. As a function of pH, then looking at changing the pH of the phenolphthalein, but now the concentration of both the phenolphthalein and the radioisotope are the same, and you can see that we do see a pH-dependent quenching of the Cherenkov radiation. We expect this to be, and it's bandwidth dependent as well as being pH dependent. This is the open spectrum here. We know the pKa is about 9.2, and you can see that we're overestimating the pKa a little bit here. The pKa seems to be around 9.7, 9.8, but in principle, the principle's there and it still works. And this is sort of what we saw last time, similar to what we saw last time, that you get the best quenching in the bandwidth where you would expect it to happen. And because this is a switchable phenomenon, we can show that we can switch the contrast back and forth. And what we did here was we took phenolphthalein in an acidic form, imaged it, added base, imaged it, added acid, and imaged it, added base. And we show that we can switch the emission of the contrast agent on and off by changing the pH. This is the decay due to the isotope decay. And again, it's bandwidth dependent with the best switching at 515 to 575, this one right here, and almost no switching at higher wavelengths. From there, we went on to try other pH indicators. This is phenol red. This is the pH indicator that is commonly used in culture medium. It has a pKa of 7.9 and shows a gradual transition from yellow to red with a lambda max of around 558. Again, the switching is possible with phenol red, but it's not as great because you have a gradual change in light emission in color intensity, unlike the phenolphthalein, which is much more dramatic. So we don't think phenol red is a particularly good candidate for this. So now these were experiments where we were just adding a pH indicator to a radioisotope. So this is what we called intermolecular quenching. And so, but from this we learned that pH indicators that absorb around 550 nanometers can selectively absorb Cherenkov radiation, and it's independent of what you put in there. It works for either sodium fluoride or F18 FDG. It's switchable, and it gives us this proof of principle for the activated Cherenkov contrast agent. But how useful is this? Can we take this on and design an intermolecular contrast agent where everything is happening in the same molecule? And we were a little worried about this because of the distance problem, and we saw Dr. Grimm discussed this, that the positrons released by F18 travel 1 to 2 millimeters before annihilation, and the velocity of the positrons decreases with distance from the emission source, and so that the sort of radius from which you can see Cherenkov radiation is about 1 millimeter. But within any kind of molecule, all of your internuclear distances are on the order of angstroms to 10 to angstroms to tens of angstroms. So would we actually be able to see an intramolecular quenching? And of course the answer is yes, or I wouldn't be standing here. And so we undertook a project to synthesize a number of F18 labeled pH indicators. We chose 3 pH indicators: phenol red, cresol red, and cresol purple. And they only differ in their structure by the number of and placement of methyl groups on the main structure. And we used an electrophilic fluorination, just a simple bubbling of F18 gas, to create both monofluorinated and difluorinated pH indicators. And we synthesized them cold and characterized their properties and found a number of interesting things. The first is that the addition of the fluorination shifts the pKa by about 0.6 pH units every time you add a fluorine. And it also shifts the absorption maximum of the basic or colored form down towards the desired region that we want. So this is actually very good. In contrast, addition of methyl groups also shifts your pKa towards the more basic region, and also shifts your basic absorbance maximum. So this kind of table here gives you a series of 9 compounds in which you can basically decide which compound you would like to use, which with wavelength and which pKa optimum. So I'm going to talk about 2. I'm going to talk about that we've found to be the most useful. One is the difluorinated phenol red, and the other is the monofluorinated cresol purple. So these are results from the difluorinated phenol red, and again, it's shifted the pKa to 6.4, the basic absorbance to 570 nanometers, but the performance of this probe is not so good. And again, even though you can see it quite clearly in the 96 well plate, and it appears in the appropriate bandwidth region, the performance is not as good because, again, you have this gradual transition in this pH indicator from yellow to red. When you go to the monofluorinated metacresol purple, this is a much better contrast agent. It has the pKa of 7.7, 582. And so 582 is right just into this bandwidth here. And you can see a very good dependence in the open filter. And this is the absorbance that you would measure just using a standard spectrophotometer. This shows that the pKa is pretty well right on 7.7. But again, we have a slight misestimation with the Cherenkov radiation for reasons that we don't quite understand yet. So to take this to an animal imaging, we wanted to image some kind of pH change in mice, and in order to get around some of the signal-to-noise limitations, we decided to image the bladder of mice where the contrast agents accumulate. So in order to do this, we took mice and we treated them with acetazolamide. Acetazolamide is a carbonic anhydrase inhibitor. It's clinically used to treat metabolic or respiratory alkalosis, and it inhibits the conversion of carbon dioxide to carbonic acid. This reaction here, so it changes the concentration of bicarbonate, and the upshot is that it leads to blood acidification and urinary alkalization. So we took mice and we injected them IP with acetazolamide for 5 days, and then injected them with the Cherenkov radiotracer and imaged the mice. Before I show you that data, I just want to remind you that this is just an image of a mouse prone and lying on his stomach and lying on his stomach and lying on his back. And this is a tumor-bearing mouse where you can see the Cherenkov radiation in the tumor, but you see the majority of Cherenkov in the bladder. Similarly, on the PET image, and when you use the selective bandwidths here, again, this is the one I showed before from a phantom, you don't actually see the theoretical change in Cherenkov radiation. You see something that looks more like this. You actually see the maximum wavelengths coming out around 620 and then cutting off, and the ones below that in the 500 range are attenuated by tissue absorption. So in vitro it looks like this, in vivo it looks like that. I wanted to show you that before I showed you the pH data. And this is the data from a mouse injected with monofluora metacresol purple. This is the PET image on the left. The PET image is on the left, and the Cherenkov image is on the right. So this is the mouse with a normal urine, and the mouse with urine of 8.5 from acetazolamide treated. Same here, 6.2, 8.5. You can see that this is a good extracellular contrast agent. About an hour after injection, you see accumulation in the liver and in the intestinal tract and a little bit in the gallbladder, and most of it ends up in the bladder. And when you look at the mouse image with the Cherenkov radiation, you can see, although there's no difference here, you can see that there's a distinct difference in the emission intensity of the mouse with the basic urine. And we know the mice have the basic urine because when they wake up, they have a P, and the P is purple, and the P is only going to be purple if the pH indicator is colored. And when we look at the different bandwidths of emission, this is the open that we saw before. We can see that, as expected, the greatest change in emission is going to be in the range where we have the maximum absorption. And you can plot that up, and you can see that, again, the greatest attenuation of the Cherenkov radiation is in the 575 to 650 nanometer region. And if you take that data and you plot it back on the pH curve, you can see that we're getting a reasonable estimate of pH from the Cherenkov data. So that's an interesting proof of principle that can be applied to any colorimetric dye, and it opens the whole possibility of imaging dyes that, functional dyes that absorb in the 600 nanometer wavelength range. And so this is the next set of experiments that we've just started. This is last week's data. We've taken MTT. MTT is a commonly used reagent in tissue culture. It's a formazine dye that you add to cells, and if the cells have functioning mitochondria, then the dye gets reduced and the dye turns purple. And so then you extract the MTT and you measure it in a plate reader, and it gives you a measure of cell viability. So this is the reaction here showing the reduction of MTT to the colored product. This is what a typical, this and this are from Wikipedia. This is what a typical plate looks like after you do an MTT assay as a function of drug concentration. And this is basically what a Cherenkov reaction looks like, taking different amounts of reduced MTT, and the concentration of reduced MT is going highest to lowest down here. And if you plot that emission as a function of dilution factor, then you can see that at higher concentrations you get maximum absorption of MTT in the appropriate wavelength, and at the other wavelengths you don't see it. So this is the proof that you can do this with any kind of dye, and this is the beginning of this is our intermolecular redox sensor, and we're now trying to synthesize the F18 labeled version, so we have an intramolecular redox sensor. Finally, this is another application. This is quenching Cherenkov radiation with red wine. This is in an attempt to develop a new method for measuring polyphenols in wine and hopefully get some sponsorship from a very expensive wine company. This is now F18 mixed with different concentrations of F18 mixed in with red wine. I know that sounds like a waste, but you only needed about 15 mils of red wine, and then you had the other 735 mils to drink. And then this is the Cherenkov emission in the absence of wine and in the presence of wine. And you can see that you get a substantially reduced Cherenkov emission with increasing concentrations of red wine. And for this red wine, we used a 2008 Turning Leaf Cabernet Sauvignon, which on cheapwineratings.com was rated as a good wine, but not sophisticated. Similarly, you can do the opposite experiment. You can change the concentration of red wine and keep the F18 concentration constant, and see that as you add increased amounts of red wine, you can increase the Cherenkov radiation accordingly. And again, it's bandwidth specific, with the maximum absorption being up here around in the red, or sorry, in the 550 to 75 to 650, getting close to the red. And it's bandwidth selective. And then finally, at some point, we'd like to take this and do some human imaging. So we've been working with Arjun Yode, and they have these diffuse optical tomography phantoms that they use to test compounds for their usefulness in human imaging. So this is just F18 in a tube that they lower into a bucket, and the bucket contains intralipid and various concentrations of India ink to simulate the scattering and absorption properties of human tissues. And we find that we can get a reasonable, if the Cherenkov phantom is within about 1.5 millimeters of the edge of the tank, we can get a reasonable imaging, and Arjun said this suggests that we could image surface tumors with as little as 1 minute acquisition time. So in conclusion, Cherenkov radiation can be selectively quenched. Inter and intramolecular quenching is possible, and this opens up a whole new world of imaging, functional imaging of chromophores in the visible region. And I'll finish with another quote from Professor Cherenkov, that there can be no doubt that the usefulness of this radiation will in the future be rapidly extended. It only took 70 years. And again, I'd like to thank all the people, and note that Julie Ciprina has a talk on Thursday afternoon on the pH agents, and I'd like to thank you for your attention.